Hello everyone, let us recall the chapter Biomolecules, and we know that this chapter is very important. Okay, so we can expect at least two questions for your NEET, one question compulsory for your Karnataka CET. Okay, so let us see the first concept of the chapter Biomolecule. Analysis of chemical composition. So we know that in the living organism or in a living tissue there will be so many organic as well as inorganic compounds, correct? Okay, so how do we analyze or how do we identify what kind of organic material is present in the living tissue? So that is the first method is given, that is trichloroacetic acid method. So you could see here trichloroacetic acid method is the first method to analyze the chemical composition in the body. So what you have to do is, like, you have to take a living tissue, okay, any tissue from the living organism, and then you need to mix it with this trichloroacetic acid, and you need to grind it, and you need to filter it. Okay, so you just filter it and you get two fractions. The one fraction which is a filtrate, so that is here, that is acid soluble pool. So acid soluble pool mainly contains the organic compounds, okay, carbon, hydrogen, oxygen, nitrogen. So all these things will be there in the acid soluble pool, whereas we get one more fraction that is called as retentate, that is acid insoluble pool, so which mainly contains inorganic compounds. Okay, so that is actually the first method. The second method to identify the inorganic compound is the ash method. Here you need to take the living tissue, take the weight of it, and after that, like, dry it, that is by heating or maybe through sunlight, whatever it is, just dry it, and after that take the weight of it and then just burn it. Okay, so after burning what we get is ash, correct? So that ash contains inorganic compounds. So here I have written organic compounds in living tissue mainly contains hydrogen, carbon, oxygen, nitrogen and sulfur, whereas inorganic compounds in living tissue mainly contains sodium, potassium, calcium, magnesium. Got it? Okay, so this is the thing. This concept, the important biomolecule present in our body and essential for our body for any physiological activity is the amino acids. The amino acid play an important role. We all know that these are the building blocks of the proteins. Obviously, if you look into the protein structure, it is made up of different types of amino acid chain, correct? Okay, so here if you look into the chemical structure, so normally, like, what you see is four groups. So for example, this is the carbon atom, and to the one side there is a carboxyl group, and to the other side there is the amino group. So amino group and carboxyl group are very important functional group which is attached to a common carbon atom. Hence this amino acid is also called as alpha amino acid. Remember what is alpha? This carbon is alpha. Okay, alpha carbon. So you could see two important functional groups attached, and we can classify the amino acid based on the type of this group, the R group. We don't know what is that R. Okay, so based on the different types of chemicals which is here attached to this carbon, which is the R group, so we can classify amino acid into different types. Okay, actually in a protein you see 20 different amino acid. Okay, and here always hydrogen will be there. Okay, so this is the basic structure, and this is now called as alpha amino acid. Amino group and carboxyl group attached to the same carbon atom. Done. Okay, so some examples. See, you have to be very thorough with the examples, because if you look into that amino acid concept, there are 9 different amino acids which are given as examples, so you have to be thorough with that. The first one is the basic amino acids, that is here the R group containing H, so that is glycine. So this amino acid is now called as glycine. Please learn the structure of it. If it is the methyl group, in the R position we have methyl group, then obviously it is alanine. If we have the hydroxyl methyl in the R position, then obviously it becomes serine. Okay, so glycine, alanine, serine are the 3 different amino acids, the basic structure. Okay, so let us just move to the next one. Again we can classify amino acid that is based on, okay, we'll keep it. Okay, so which is based on the number of amino group and number of carboxylic group. It is classified into 3 types. Okay, so how many amino groups are present in one particular amino acid, how many carboxyl groups are present in one particular amino acid, like that. Okay, so it can be classified into 3 types. Here I will not go into detail. I have made a video on this. Okay, so if you want, I'll share that. So acidic amino acid. So here example given is glutamic acid. Glutamic acid is a good example for acetic amino acid, very important for NEET or CET, okay, could be asked. Basic amino acid, lysine. Neutral amino acid, the valine. Okay, so glutamic acid, lysine, valine are the example of acidic, basic, neutral amino acids respectively. Aromatic amino acid, very simple. In the amino acid structure, if the amino acid contains the ring structure, the aromatic ring, then obviously that is aromatic amino acid. There are three types: tyrosine, phenylalanine, tryptophan. So these are the three important examples for aromatic amino acid. Please remember, could be asked, very important. So total 9 amino acids you need to remember. Last one and very important one, zwitterion. At a particular pH, the amino group as well as the carboxyl group can be ionized. Got it? They get the charge. So you could see that it's already given in the NCERT textbook. Please remember this, and yeah, being a chemistry student, and it should be easy for you. Okay, here it is positive, positive, negative. See? Okay, so this is zwitterion state. Got it? At a particular pH, amino group as well as the carboxyl group can be ionized. Okay, so that's actually the thing. Now this is another important concept. Okay, lipids. What are lipids? Lipids are nothing but fatty acids. Sometimes, along with the lipids you see, along with the fatty acids you see glycerol also. Okay, so based on the number of glycerol we can classify the lipids as monoglycerides, diglycerides, triglycerides. Got it? But remember, fatty acid plus glycerol. Okay, so but basically lipids are nothing but fatty acid. Okay, a fatty acid has a carboxyl group. Okay, compulsory thing is they should have this group, carboxylic group, okay, attached to an R group. Got it? Okay, this R group can be any carbon compound. Okay, so R group could be methyl. That's what here. It could be methyl, it could be ethyl, or it could be any other compound, but it should contains carbon. The number of carbon atoms in that group could be range from 1 to 19, maximum 19. It should not exceed more than 19. Got it? Okay. So this one, please note this, very, very important. Arachidonic acid. Okay, it has got 20 carbon atom, including the carboxylic carbon. That means COH is compulsory, and along with that you see 19C. That's it, total 20. So 19 plus 1, 20. Okay, this so arachidonic acid, remember that. Whereas the palmitic acid, it is also called as fatty acid. Palmitic acid is nothing but fatty acid. So this is the structure. Please note this point. If you count the total number of carbon, so 1 here, so 14, 15, now 16. Total 16 carbon atoms, including carboxylic group. Okay, so this is very, very important. Don't forget, arachidonic acid 20, whereas palmitic acid 16. So remember this. Then the glycerol, as I said earlier, glycerol sometimes actually found along with the fatty acids with a bond. The bond is called as the ester bond. So this is written here, glycerol plus fatty acids, that is because of ester bond. Okay, so again now we can classify the lipids as three types based on the number of glycerol attached to the fatty acid, that is monoglycerides, diglycerides, triglycerides. It's not mentioned here. See, glycerol, simply trihydroxypropane. See, you could see the structure of a glycerol. Okay, this is glycerol. Okay, trihydroxypropane. Done. Okay, simple. Next, we know that oils, whatever the oils you take, sunflower oil, okay, so they will remain as oils itself during winter season because they have low melting point. I'm stressing this point, very important. Then this one, the lecithin. You know that the plasma membrane of any living organism is mainly made up of lipid bilayer. But what is that lipid? The lipid is nothing but lecithin. So that is found in plasma membrane. Remember, lecithin is the main lipid which is found inside the plasma membrane, which makes the lipid bilayer. Got it? So structure of the lecithin is given in your NCERT textbook. So please note this. No need to learn this, remember, but just see this, and you should be able to identify it if it is asked in the examination. Got it? This is lecithin. Okay, so no need to learn the chemical structure of all this stuff whatever I said, but it could be asked in the option part, so you should be able to identify that. That's it. Now we know about nitrogenous bases, and we all know mainly there are 5 nitrogenous bases, 4 in DNA, 4 in RNA. Okay, but thymine is the only nitrogenous base found in the DNA, whereas uracil is found only in the RNA. You know that. Nitrogenous bases: adenine, guanine, cytosine, thymine, uracil. Okay. See, this adenine always found with, for example, we know that there are 2 types of structures, that is purines. Purines mainly contains adenine and guanine. Pyrimidines contains cytosine, thymine, uracil. Correct. So adenine and guanine is heterocyclic. What is heterocyclic? They have two rings. So they have two rings, whereas pyrimidine is only having only one ring. Got it? So that's very important. You should be able to identify that. If heterocyclic ring is given and you need, if it is asked like what is it, the structure, adenine or guanine, cytosine, thymine, you should be able to identify it. Okay. So adenine, when it is found along with the ribose sugar, we know that there is a sugar called as ribose sugar. Okay. So adenine, the nitrogenous base, which when it is attached to the ribose sugar with the help of N glycosidic bond, it makes a structure called as nucleoside. So adenine plus sugar, adenosine; guanine plus sugar, guanosine; cytosine plus sugar, cytidine; thymine plus sugar, thymidine; uracil plus sugar, uridine. So nucleoside. So this is nucleoside. You know about it. What is nucleotide? When the nucleoside is attached to a phosphate group or phosphoric acid. Remember, phosphoric acid gives acidic character to the DNA or RNA. Remember that, and it also gives negative charge to the DNA. Okay, whatever it is, when nucleoside binds to the nucleotide, the phosphate group, it makes a structure called as nucleotide. So nucleoside plus phosphate is with the help of phosphodiester bond. In simply, we can call it as ester bond. Yeah, we call it as phosphodiester bond because there is a phosphate group. Correct. Easy. So the nucleotide of adenine and this one, sugar, when it is attached to a phosphoric acid, that makes nucleotide. Adenyllic acid for adenine, guanine, guanylic acid, cytidyllic acid, thymidyllic acid, uridylic acid. Easy. Okay. So now this is very important. You could expect at least one question from this bonds part. Okay, linkages between nitrogenous base and sugar, N glycosidic bond. Between nucleoside and phosphate group, ester bonds. What kind of ester bonds? Phosphodiester bonds. Note, in DNA, adenine, guanine, cytosine, thymine is present. RNA, adenine, guanine, cytosine, uracil is present. That means RNA, thymine is absent, whereas in DNA, uracil is absent. That's it. Purines, adenine, guanine, two rings are there, hence it is heterocyclic. Pyrimidines, cytosine, thymine, uracil. How many rings? Only one ring. So it should be easy. So between two amino acids, what type of bond is present? Peptide bonds. Ultimately, many amino acids are linked by peptide bond and ultimately make a polypeptide chain, and polypeptide chain is nothing but protein. So primary and secondary metabolites, this is very, very important. See, in any living organisms, there are so many chemicals which are produced as a result of metabolism, correct? So some chemicals which are produced by the organisms, they are important for their physiological activities, for their metabolic activities. So they are called as primary metabolite. Whatever the chemical which is produced by the organism, if it is used in a metabolic process or if it is used in a physiological process, then that is called as primary metabolites. Okay. So, whereas sometimes the chemical which is actually produced by the living organism doesn't play any role in their physiological activity, so then they are called as secondary metabolite. Remember, produced by the living organism but doesn't play any role in their physiological process or metabolic process. They are waste actually for that living organism, but for human beings it could be useful. For example, see, pigments. There are so many pigments which are secondary metabolite. Example for secondary metabolite, you have to be thorough with this, you have to mug up, I'm telling you. Carotenoids, anthocyanin. These are the pigments. You know that anthocyanin is the pigment present in the beetroot. Okay. Alkaloids, morphine, codeine. Terpenoids, monoterpenes, diterpenes. Essential oils, lemongrass oil. Toxins, abrin, ricin. Remember that. Lectins, concanavalin A. So pronouncing is difficult. Concanavalin A. Okay, so this one is important. I saw this somewhere else in the competitive examination. Drugs, vinblastine and curcumin. Okay, polymeric substances, rubber, gums and cellulose. Okay, so this is actually important thing. Lipids are normally under a micromolecule because they have low molecular weight. That is 800 Daltons. Just note this point. That's enough. Okay. So we go further. Protein, very important. So we know that protein is nothing but heteropolymer. What is the meaning of hetero? Different. It is not homopolymer. For example, let us take glycine. So glycine, many amino acids, but the same amino acid. They are linked by peptide bond. they cannot make protein. If glycine, serine, alanine, tryptophan, all these different types of amino acid, they linked by peptide bond and make a long chain, it could make a protein. So that's heteropolymer. So long chain of protein which is made up of different types of amino acid, but a protein can contain only 20 amino acids, but the number of individual amino acids can be more. Okay, so it could be like 100 amino acids, but if you just see them, it should be within 20. Okay, essential amino acids. What is the meaning of essential? Say body cannot produce these amino acids, but it's required for our physiological process, compulsory. We need to take the amino acids in the form of food. Non-essential amino acid, this means like body actually produces it, so no need to take in the form of food. That's it. Okay, body can produce it. Remember these two. Some examples, this is very important, normally asked in match the following. Protein and functions, very important, don't forget. Collagen, we know that intercellular ground substance, and we know that this is the protein is abundant in living organisms. Your body mainly contains collagen. Okay, so it is intercellular ground substance. Okay, that gives a proper structure to the human beings or any animal. Trypsin, we know that trypsin is an enzyme. Insulin, hormone. Antibody fights infectious agents. Correct? Okay, for immunity. Receptors, there are so many receptors like for smell, taste, and even hormones also. For hormones also receptors will be there. Then GLUT4, remember that these are the receptors which enables the glucose transport into the cell, normally seen like for example this is the cell, you could see the receptor that is called as GLUT4. So GLUT4 actually accept the glucose which is present in the bloodstream, and that will be entering into the cell with the help of GLUT4. That's it. Okay, so this is very important. We are done with that. We'll go to the next sheet. Yeah. Okay, so collagen. We know that collagen is one important protein that is most abundant protein in animals. Rubisco, you know what is Rubisco? Correct. Ribulose, bisphospho, carboxylase oxygenase enzyme, which is most abundant protein in the biosphere. In the world, it's the most abundant protein, and in the plant world, it is most abundant, obviously. Okay, so protein structure, there are actually 4 structures in the protein: primary structure, secondary structure, and tertiary and quaternary. Please note this point, very important. Primary structure, it doesn't exist actually, but still the scientist actually made one structure for a primary structure. Okay, primary means it's like a long linear thread. Okay, it's nothing but a line like structure. Okay, it has got 2 ends. The starting amino acid in the polypeptide chain contains is called as N terminal. The last amino acid in that chain is called as C terminal, NC. N terminal first amino acid, C terminal last amino acid. Remember that the first amino acid is N terminal, last amino acid is C terminal. Got it? Primary structure is like a line. This structure doesn't exist. Note this point. Okay. We'll go to the secondary structure. This is very important for your competitive part because it mainly contains 2 important parts: alpha helix and beta plated sheet. Okay, secondary structure, normally you see the polypeptide chain which is right-handedly coiled, you know, DNA structure which is also right-handedly coiled. Correct? Okay, so there are 2 types. Alpha helix. Alpha helix is for the polypeptide chain if it is only one. If there is only one polypeptide chain, and if it is right-handedly coiled, and this is the coil, and each coil is linked by the hydrogen bond. That is alpha helix. Okay, it's written single chain arranged like a coil, and they are linked by the hydrogen bond. I think I missed it somewhere else. Yeah. Okay. So here, sorry, no hydrogen bond. So yeah, hydrogen bond is present. Yes, I missed it. Whereas the beta plated sheet here, this is bit upgraded. If protein has more than 2 polypeptide or 3 polypeptide chain, obviously they are all right-handedly coiled, but they are actually linked to one another by a plate-like structure. So you could see that. So this is one polypeptide, this is the other polypeptide chain. So here the linkage is mainly brought by hydrogen bonds. Remember hydrogen bonds, that's more than sufficient actually. Commonly asked what type of bond is present in secondary structure or beta plated sheet, that is hydrogen bond. Simple. Okay. Third, tertiary structure. See, it's a long thread of polypeptide chain which is irregularly arranged like this, like looks like a woolen ball. Okay, remember that. And even 3D structure also looks like kind of this kind of it. Okay, so and here what we have is hydrogen disulfide bond which actually links it. So between these, here within this you could see so many bonds, that is hydrogen disulfide bond. Please remember, tertiary structure contains hydrogen disulfide bond. Okay, So we are done with that. We are going to the next sheet. Last one, the quaternary structure of the protein. Very simple. If you remember the antibody, so what is this? Heavy chain, correct? How many are there? Two are there. What is this? Light chains. How many are there? Two are there. So how many subunits of polypeptide chains are there? 1, 2, 3, 4, correct? So that is quaternary structure. Even if you look into the hemoglobin structure, see, hemoglobin is made up of 2 alpha chains and 2 beta chains, that is polypeptide chains. Alpha polypeptide, beta beta, correct? So this is the thing. So 4 subunits of proteins will be there, and even here also you could see that linkage is mainly brought by disulfide linkages, correct? So what kind of linkage? Disulfide linkages will be there. Okay, fine. So that's all about the quaternary structure. Just remember, hemoglobin and antibody is a very good example for the quaternary structure. Polysaccharide, normally cellulose, glycogen are very good examples for polysaccharides. Okay, these are the long chain of sugars. Here it could be homopolymer or it could be heteropolymer, both are seen. Got it? For example, you take cellulose. Cellulose mainly contains only like glucose, correct? Glucose. Whereas even if you take the glycogen also made up of glucose units, okay, that is monomeric units. Long chain of sugar, fructose is the only polysaccharide which is very common in this one, what you call fruits. Got it? Okay, actually it's not a polysaccharide. Okay, fine. Monosaccharide, but it's very commonly seen in fruits. Glycosidic bond, okay, so this is very important. In carbohydrates you see only glycosidic bonds, got it, between two individual monosaccharide units. Okay, cellulose, homopolymer of glucose units. As I said, like many G's, that is glucose units, ultimately make cellulose. Starch, storehouse of energy in plants, we know that. Glycogen, storehouse of energy in animals, we know that. Inulin, it is not insulin, inulin, it is homopolymer of fructose. It may be asked actually. So inulin is homopolymer of fructose, glucose, or some other thing, starch, whatever it is. So be thorough with this. Inulin is fructose. Many fructose units makes the inulin structure. This page is sheet is very important. Okay, so glycogen. So we know that glycogen is mainly made up of many units of glucose. Many glucose attached to one another by N-glycosidic linkage and ultimately make a long chain called as glycogen. It has two ends. Got it? So normally if you take carbohydrates, carbohydrate has two ends actually. So the starting end is reducing end, the last end is called as non-reducing end. That's what written. Right hand side is reducing end. Right hand means for that. See, this is for us like left hand, but it is right hand. So that is reducing end, and this one is non-reducing end. That's it. Okay. So here if you look into the structure, see, this is now a linear structure. So this is one glucose, second glucose, third glucose, fourth glucose. Many glucose will be there, and see, if you just clearly look, see, magnification, see, I have magnified this particular structure. See, this is the first carbon, second carbon, third carbon, fourth one, fifth one, and sixth one will be somewhere else here, not here. Got it? Okay. So 1, 2, 3, 4, 5, 6. So this is the thing actually. So here the linkage between, if you look into this linear chain of the glucose, that is glycogen, between the first carbon of the first glucose and fourth carbon of the second glucose, the linkage is there. What kind of linkage? N-glycosidic linkage. So this linkage is called as beta 1,4 glycosidic linkage. Remember that in a linear chain you see beta, not alpha, beta 1,4. Why 1,4? 1 here and 4 here, 1,4, and this linkage is N-glycosidic linkage. Got it? Easy? Okay. So this is the thing, but this is the linear chain. So we know that this is 1 and 4 ultimately make beta 1,4 N-glycosidic linkage, N-glycosidic, everything is common. But here to the sixth carbon atom, another chain of the glucose monomeric chain is attached. See? So this is the linear chain, and this is the branches. You could see so many branches, but these branches are attached to the sixth carbon of this main chain. Got it? Okay. So if you look here to the sixth one, the first carbon of the first glucose unit is attached of another chain, correct? So you could see here. So this is 1 and 6. So if you look into the structure of glycogen, you could see between sixth carbon atom of one glucose and first carbon of another glucose, like carbon of another glucose chain, which is linked by alpha 1,6 glycosidic linkage. Here 1 and 6, correct? But this is alpha 1,6 glycosidic linkage. Done. Okay, so this is the thing. I made a mistake. I was telling N. So N cannot be seen here because it's purely carbohydrate. N can be seen because if one purine or pyrimidine which is attached to the ribose sugar. Okay, that is N, nitrogen. Okay, so here nitrogen is absent, sorry for that. So it is only glycosidic linkage. Okay. Yeah, we are done. Last sheet. And see, this is very important. If you look into the starch is actually helically coiled. Okay, they are helically coiled, and because of this structure, they can hold the iodine. Got it? So obviously, when starch binds to iodine with a helical structure, it turns blue. This is very important. Whereas cellulose is linear. Correct? It's a long chain, so it doesn't have any helical structures, so it cannot hold iodine. So it cannot turn blue. That's it. Reducing sugars, this is, please note this, it's not given in NCERT, but there is a chance in NEET. So name some reducing sugars: glucose, fructose, maltose and lactose. Okay, reducing sugars. Non-reducing sugars, example, only one is given, that is sucrose. Just remember that, sucrose. What is a living state? Living state is nothing but, remember, non-equilibrium steady state to be able to perform work. Non-equilibrium. So what is the meaning of non-equilibrium? So the metabolic rate should not match with the living state, okay, or the steady state. So that is non-equilibrium. So metabolism always maintains the living state. So it's because of like what kind of reaction is occurring inside your body. So based on that, your other organ starts to work. So metabolism is equal to living state. So metabolism and living state are synonymous. So this is very, very important. Done. Okay. So we are done with that. Yes. So the last concept of this chapter is the enzymes. Correct. So I have to take another session because enzyme concept is not that easy as whatever I have discussed so far. It requires extra session, that is board teaching is required for that, or maybe like a handwritten. Right, I have to write and explain it. Okay, so I am going to merge structural organization in animals and enzyme concept in one session. Maybe I need some days. Okay, but I'm going to cover it. Okay, don't worry about it. Enzyme concept. So fine. So whatever we have discussed is very, very important. That is more than sufficient. Also, everything is covered. Almost everything is covered. Okay, if you watch the video, then it's good. Okay, if you don't, means you are going to miss so many things. Okay, so chalo then. Take care.